Hi, good morning, everyone, and a warm welcome to all of you for joining us in this Twist webinar on the topic, Capabilities of the new Twist NGS Methylation Detection System. My name is Eileen, and I will be your host and moderator for today. I'm very excited to have you join us in this webinar. So in this webinar, you will hear about the benefits of the Twist pre-capture conversion probe design approach and how the Twist NGS methylation detection system workflow leads to increased confidence in methylated base calls. So before I introduce to you our speaker, just a few housekeeping items. All lines will be muted during the webinar. We will answer all questions at the end of the presentation. If you have a question, please submit it in the Q&A box rather than through the chat box. And finally, following the presentation, if you could please take 1 minute to take a brief survey, we would greatly appreciate your feedback. And now, it gives me great pleasure to introduce to you our guest speaker, Dr. Ananth Bommakanti. Dr. Bommakanti earned his Ph.D. in molecular cellular biology from the University of Maryland, Baltimore. He has been with Twist Bioscience for the past 2 years as our bioinformatics and NGS field application scientist, helping customers accelerate today's NGS-based research using Twist range of NGS products. So without further ado, may I invite Dr. Bommakanti to kick off this presentation. Dr. Bommakanti, please.
Hi, good morning, everyone, or maybe good afternoon. And thanks a lot, Ely, for such a nice introduction. So I'm Ananth, and I'm the NGS application scientist for the KOSEA region. For all those who are new up here, let me start with a quick introduction about Twist Bioscience, what we do and how we do it, and why we are better compared to our competitors. So in short, Twist Bioscience is a synthetic biology company, and then we make DNA, and we do our job exceptionally well. So we synthesize our DNA using a disruptive throughput technology that uses microfluidics and silicon platform, and synthesizes the DNA using the same phosphoramidite chemistry. And so compared to our competitors up there who use a 96 well plate or a 384 well plate, we use the silicon wafer. And on this wafer, we can print up to 1 million oligos in a given 36-hour time frame. So we can synthesize very high quality oligos with a rapid turnaround time. And these oligos form the main basis of our verticals, which are the genes, oligopools, and libraries, which form the core offering of our SynBio space. And then we have the futuristic DNA data storage offering. And then we recently ventured into biopharma space. And we are having products that are helping currently with the COVID scenario that we are all are facing. So for today, let me talk more about the NGS offering, and more specifically about the methylation detection system. So why we are better compared to our competitors? The first thing is we provide exceptional performance, right? So this comes from using double-stranded DNA probes that capture the targets with very high efficiency and leads to exceptional uniformity, which I would say is the best in the class. And then the second thing is we NGS QC all our probes before sending it for the enrichment purposes. This way you can avoid false negatives, and then you can avoid the probe dropout. And because there won't be any probe dropout, you can rule out false negatives. And we also offer great flexibility in this sense. First thing, we offer off-the-shelf products like Exome, which you can directly use with our kit. Or we have the capability to print and rapidly optimize small panels small to very large panels, and like a small panel like a mitochondrial DNA panel or an MRD panel, and large panels like clinical exomes that the customers can customize according to their needs. And with this, we also have modular kits where you can use it for all your needs, and then you can rapidly customize the kits, and then depending upon the your experimental needs, you can customize them. And all these things will lead to maximize sequencing efficiency, right? Providing the most bang for your buck. And then irrespective of what kind of sequencing you're using, irrespective of what kind of sequencer you're using, you can actually get amazing cost efficiency, either it be a benchtop sequencer or a production level sequencer. So before going into the main, talking about our new exceptional offering, let me quickly talk about a few metrics that will bring everyone of us onto the same page, and then also show why we have such great products for you to offer. So unlike whole genome sequencing, the hybridization target capture in general has a few fallacies where there is a lot of bias that arises during hybridization capture. There is because of the expansion of the probe pools, or because of the inherent nature of the DNA that is being captured. Right. And for example, if you want to capture a target that has 3 exons, which you can see up here, when you perform the hybridization capture efficiency, you can end up with targets that are covered at the depth that you are intended, or there'll be targets that have higher coverage than what you want, and there'll be targets that are covered at significantly lower levels than you are aiming for. And again, there are these reads that don't belong to the capture area that you are looking at, which are usually the off-target reads. So all these 3, the over-sequence regions, under-sequence regions, and then also the off-target effects will eat away your sequencing space. This is wastage, this is wasted data, and then actually it will be costing, it adds to your sequencing cost. So at Twist, the main aim is how we can repurpose these additional reads so that you'll be getting uniform coverage all throughout the panel that you are looking at. And we achieve this using multiple ways. The first thing that we use is the uniform oligo pool that we use for capturing the targets that you're working with. Right. So when you synthesize these oligo pools on the silicon wafer, because we are using a phosphoramidite chemistry, the probe has a very good uniform GC distribution, and there are not much bias in the oligo pool. But the pool that is produced on the silicon wafer is very low in concentration. It's less than 1 femtomole. And this needs to be amplified so that it can reach to a quantity which can be used for hybridization capture. So the usual PCR amplification has so much bias in them. And when you amplify this oligo pool that is produced from, that is synthesized on silicon wafer, will end up having huge GC distribution bias that you can see in this graph, right? And when you use this oligo pool for capturing your target region, you'll always end up with a sequencing result that has so much of unevenness or very little uniformity. But and this is one of the main reasons you get a very crappy results, right? If you're using a garbage oligo pool in, you get bad results. But at Twist, we have a proprietary amplification method where we ensure that the GC distribution that is there during the synthesis of the oligos is actually replicated in the amplified oligo pool. And when you use this, uniformly distributed GC binning oligo pool, you will end up with a very uniform capture result. This is one of the first steps where we take, that leads to the uniformity that we achieve. And usually this uniformity is measured as fold-80 base penalty, and everyone who is working with the hybridization capture knows this particular metric. But in short, this is this metric essentially tells us how much additional sequencing is required to reach the depth that you are aiming for. And closer to one, the metric, the better the uniformity is. And you can see up here, compared to the vendors, compared to the other competitors out there, we achieve a uniformity that is very close to one. And this is one of the main reasons you have amazing sequencing cost savings because you are sequencing little and putting more number of samples on the sequencer. And the other aspect, which I'm not showing up here, is we actually GC bin the pools, and we kind of adjust the stoichiometry of the probe pools that we send to you. That is one of the other reasons why you get this kind of uniformity when you use twist capture pools for your target capture. The second aspect of our workflow is we use double-stranded DNA probes, and when you use double-stranded DNA probes, essentially you're capturing both the strands of the DNA. This actually plays a role when mutations are present only on one particular strand, or if you're working with degraded or damaged DNA samples. And one of the main advantages of using this double-stranded DNA probes is you get 4 to 6 fold more unique molecules. As you can see up here in this graph, compared to the other vendors, we get 4 to 6 fold more unique molecules in the library preparation stage, and in the which eventually trickles down to the capture stage, right? And this uniqueness, this rich library complexity directly equates to the duplication rates that we achieve, right? And so our duplication rates are usually less than 2%, which is the industry standard, right? And this metric also again equates to the same thing, where you are discarding low amount of reads due to your duplicates. So you're having actually more data to analyze, and then which again is equates to the cost savings during sequencing. So the third aspect where Twist excels is the custom panel design process, right? And we have advanced design algorithms, and we call this as the learning loop, where we can design the panel for you and build it very quickly. And then once you test the panel, we can learn from the data that we get from the panel results, and then we can re-optimize the design if it is necessary. And this entire process takes place very rapidly. Within 6 weeks, you can have the data on for optimization, and you'll have, you can go from your pilot to production stage really fast. This is partly because of our capability to print high quality oligos in a very rapid time, and also because we have these amazing test, build, learn cycle. And these examples of the panels that we have designed straight off of the designer tool without any optimization. You can see that smaller panels, which is the mitochondrial panel, which is close to 17 kb, or exploratory cancer panel, which is around 13 MB, and our exome is around 37 MB, right? So irrespective of the panel size, when they come off of the design, they perform exceptionally well. You can see how tight the uniformity is irrespective of the panel size up here. And then the duplication rates are also the same thing. They don't really get affected by the panel size. They are very small. And this, this rapid optimization is one of the main stands for us. And then when you combine all these three metrics with seamless workflows that we have created, where we can provide end-to-end solutions, right? We have complete kits where you can prepare your libraries and then use the Twist enrichment reagents. Or if you have some existing libraries that have been already made with what do you call some libraries that are compatible with the Illumina platform, you can always use just the enrichment kits, which have universal blockers that are compatible with any library preparation out there. So depending upon experimental needs, depending upon your research needs, you can actually customize it and get an end-to-end solution with Twist. So to extend the same performance flexibility that we achieve with our whole exome sequencing and custom target enrichment thing, scientists at Twist Bioscience developed an end-to-end workflow which is pretty advanced and involves a targeting sequencing method for the methylation detection, and that actually provides the best, as I'll show you in later slide, it produces the best uniformity and sensitivity. So epigenetics has become an important area of research, especially in the areas like liquid biopsy or early cancer screening, right? And areas of neurobiology, or even just for understanding cellular growth and development. Methylation is one of the key areas of the epigenetics that is being extensively studied recently. And the Twist targeted methylation workflow, we introduce a solution that produces highly complex libraries, gives you exceptional performance, uniformity, and sensitivity. And again, with our custom design probabilities, you can have a rapid panel design and optimization with flexibility in the design that you want for your target areas. And then also, we came up with the end-to-end workflow that involves innovative enzymatic conversion process that is very gentle on the DNA that you're going to use. So scientists at Twist, so to bring this workflow to you, the scientists at Twist have actually gone back to the basics and came up with this pre-capture conversion process. So saying that, so essentially you modify the DNA that you're working with and then capture the regions of interest. So unlike post-capture conversion, what exactly, where you use very high amount of DNA, and even after using that much high amount of DNA, you necessarily need not have good library complexity. But the pre-capture conversion process that we use gives you a very good library complexity, and then because we capture a high number of unique molecules, and also at the same time you can use very low input DNA amount, which is a very key aspect, especially when you're working with samples like cell-free DNA or FFPE samples, right? Lower input DNA is something that a situation that we often encounter with. And also because it's not a chemical process, it's safe and pretty easy to work with. It's less cumbersome compared to the post-capture conversion process. We'll see those aspects in the next slides. But one thing that I want to emphasize is the pre-capture conversion process has one major hurdle that we had to come up with, right? Because when you're converting the reference target, you're actually modifying the target pretty significantly. And even with a small target size, the combinations of methylated, unmethylated regions can be easily from few dozen to 1,000, right? And then we have to design probes so that all these modifications are taken into considerations, and then all the probes that we design should match exactly with the modifications that are being done on the target. So if it was not, I would say it as pre-Twist Bioscience and post Twist Bioscience era, right? If not for our disruptive technology, coming up with so many probes that would target all these modifications would be next to impossible, right? So we rely on our super silicon, super technology where we use a silicon platform for printing millions of these oligos. So and then along with this printing capability, we came up with the advanced AI-based design platforms. So what it does is it actually designs probes to both the sense and anti-sense of the target regions, and then also both the unmethylated regions and the methylated regions of the target region. So essentially, each target will end up with 8 distinct probes. And depending upon the combinations, these number of probes will vary drastically. And because we are using 8 different probes to capture all these things, you will end up with very rich library complexity, and that will eventually lead to very sensitive target enrichment. And so now we have this really good probe technology, and we want to complement this probe technology with the library preparation, right? So Twist and New England Biolabs have partnered to produce the end-to-end solution into the market. So this NEBNext EM-Seq for Twist, Twist targeting methylation sequencing involves a ligation-based library preparation module, and then a unique enzymatic conversion module where you'll modify the reference target or the target that you're interested in. And then this entire kit is optimized for using with the Twist target enrichment workflow, where we use a optimized fast hybridization system that reduces the entire time of your enrichment. And then we have a unique methylation enhancer added to this system. This reduces the off-target reads pretty drastically. And when this entire system is combined with our pre-optimized custom design panels, this leads to best-in-the-class end-to-end solution available for the methylation detection. So the just a quick view about the workflow for the NEBNext EM-Seq system for targeted methylation. It's a very simple and straightforward workflow. It involves a library preparation where we mechanically fragment the DNA and repair a tailor, and then add the methylation specific adapters to it. And then it involves steps that would enzymatically convert the target for methylation. And then we have the libraries that are good for enriching using the Twist target enrichment system, which is a quick 7-hour protocol. And once you have it, if you add the 32 hours of sequencing time, within say around 3 to 4 days, you'll have the entire data in your hands that is ready for analyzing the sequencing. So it's a very efficient, fast, and swift workflow, very smooth and rapid workflow that will help you with your detection systems. So let's just quickly look at the data pertaining to various steps in the workflow. Starting with the library preparation using the NEBNext Enzymatic Methyl-Seq kit. So the traditional bisulfite sequencing, traditionally the methylations are detected using the bisulfite conversion method, or the whole genome bisulfite conversion of bisulfite sequencing, right? Where sodium bisulfite, a chemical, is used for modifying all the unmethylated cytosines to uracil, which are eventually converted to the thymines. This is industry standard, and it has a very good accuracy. But saying this is a chemical process. It is harsh, and then it actually damages the DNA. It causes DNA strand breakage, DNA pyramidization, depyramidization, and the efficiency of conversion is not that uniform. It's not uniform, and it's not even consistent as well. But unlike the traditional method, the EM-Seq kit actually uses an enzymatic method. which uses two enzymes for converting the DNA, resulting in the same kind of target that you get with the sodium bisulfite method. Let's look a little bit deeper into the enzymatic method. So as I said, the NEB kit uses enzymatic method, uses enzymes for achieving the same result that you get with the bisulfite sequencing. So the first step involves protecting the protecting the methylated and the hydroxymethylated cytosines. And then once you get the DNAs where these modified cytosines are protected, we denature it. And then we use the second enzyme where all the unmodified cytosines are converted to uracil. And subsequent PCR steps will convert this uracil to thymine. So you end up with the same product that you get with the bisulfite sequencing, and using the similar bioinformatic pipelines, you can analyze for the cytosine modifications. So because we are using an enzymatic method, this has very superior sensitivity, and then it gives you, there is no DNA damage per se, and it gives better mapping efficiency. It's highly uniform. And then it actually detects more CPGs compared to the bisulfite sequencing method. So in the subsequent slides, I'll show you the data pertaining to these metrics up here. So first thing is, along with the kit, we include along with the kit, we include both two controls. One is unmethylated lambda DNA, and the other one is the CpG methylated POC19 DNA. So these can be used for measuring the conversion efficiencies. And here in this slide, you can see that both the controls perform exceptionally well. When you measure the conversion efficiencies for the unmethylated control and methylated control, both for the methylated, the unmethylated control and the methylated control, you get more than 99.5% of the conversion efficiency. And with the unmethylated lambda, you get less than 5% measured methylations. And for the methylated CpG, you get more than 95.5%. So this shows that the conversion efficiency match the expected levels of these controls. And then that the conversion process is complete, and then you won't get any false positive, or at least the false positives are kept to a minimum. Now, if we look at the actual data, the enzymatic method provides very efficient conversion, like I showed in the previous slide. So here in the left, the graph to the left, you can see that the x-axis shows the conversion rate, and compared to the bisulfite sequencing, bisulfite conversion, you can see that the enzymatic method is more efficient, right? And this, when you actually look at the amount of DNA that you get after your library preparation, with bisulfite method, without any optimization, you get less than 10 nanograms of your library after the conversion process. And you can optimize it to increase this particular concentration. But with the enzymatic method, you can see straight off without any kind of optimization, you end up with more than 110 nanograms. This actually shows that the amplifiable molecules that are more with the enzymatic conversion compared to the bisulfite conversion, right? So this directly shows how rich the library complexity will be once you prepare your libraries using the enzymatic conversion process. So the second aspect is the DNA damage. Like I mentioned previously, because bisulfite method is a chemical process, it does damage your DNA, right? And this is pretty much evident when you're using cfDNA as your starting material. So everyone who works with cfDNA know that when you follow the traces of the cfDNA, you end up with these high molecular weight fragments that are a result of the nucleotide positioning, right? And then when you use bisulfite conversion method for converting your circulatory to cell-free DNA, you can actually see that these high molecular weight fragments are actually lost. But when you're using enzymatic method, these nucleoside positioning markers are pretty well preserved. And recent studies have shown that these markers are pretty important, as they differ in both cancerous states and non-cancerous states. So this shows that the bisulfite conversion method actually is more harsher and leads to DNA breakage compared to the enzymatic method. So not only just being less harsh on the being gentler on the input DNA, you can see that the conversion efficiency itself is very good compared to the bisulfite method. So here in this graph on left, you can see when you compare the cytosine conversions between the enzymatic method and the bisulfite method, there is a very nice correlation between both the methods. And then the conversion is, the enzymatic conversion is very good without minimal bias, depending on the DNA that you're using, right? And also, if you look, it's not only it converts the cytosines very efficiently, but actually it converts, converts 15% more cytosines. So here you can see that compared to the bisulfite sequencing, which is in lighter green up here, you can see that the enzymatic conversion actually converts 15% more CpGs than the bisulfite conversion method. And we can look at more data in the subsequent slides. But so now, so to summarize, the EM-Seq provides improved results. It has better sensitivity compared to the bisulfite sequencing, and you can use less amount of DNA for getting the same amount of data that you use with your bisulfite sequencing. And this obviously will lead to good sequencing cost, right? And you can see how these results are enhanced when you use the target enrichment with Twist, right? So let's move into the second segment, which is the enrichment using the Twist target enrichment workflow. So just to recap, I explained you that we use a pre-capture conversion method, right, where the reference target is modified first, and then using the Twist probes, we capture it. And again, this gives a very good library complexity, and you can get away with lower DNA input amount compared to the post-capture conversion modules. And just to show how complex the library becomes after the conversion, here you can see that if first thing, the genomic sequence are modified. This is the regular genomic sequence that you can see here. And once the conversion happens, the complexity actually reduces between the fully methylated and fully unmethylated sequences. The sequence complexity is notably reduced after the modification of the reference genome. And the GC percentage obviously will change because we are converting all the non-modified Cs to thymines, right? So this poses multiple issues up here. First thing, the repetitiveness of the DNA will increase, and also this will lead to higher off-target effects, right? Because the GC percentage is changing. And then the entire hybridization kinetics needs to be reevaluated when we are using this kind of system where we are modifying the DNA and then capturing it. And as I said, Twist achieves this using multiple segments. Every aspect of the workflow has been optimized to get the results that we are looking at. So first thing is the probe design. As I mentioned before, we use advanced algorithms where we capture both the sense and the antisense strands, And also we capture the methylated and unmethylated regions of the target. And we use machine learning algorithms to fine tune this entire design process per se. So what it does is it takes all these various combinations that result after the modification, and then we put it through a design, and then it does two things. The first thing is it actually optimizes the target that we are looking at. When I say it optimizes the target, it might remove some regions that has in a lower complexity or higher repetitive regions. So here you can see that the default panel size without optimization is 1.4 MB, 49 MB. And once we optimize the target where we remove some regions that might cause higher off-target effects, the target size is not changed drastically. It just becomes 1.48 MB, right? But you can see the significant change in the off-target effects that we achieve after this target optimization. So once, and then the 30x coverage also improves compared to the non-optimized or the default target and the optimized target. But it doesn't really affect the uniformity, which and the duplication rates that you get after the target optimization. And the second thing that it does is it actually adds a stringency. So depending upon your, what the experimental needs are, it does, we design the panel probes in such a way that it follows three levels of stringency, where we can have low stringency or, medium or high stringency. So if you see that as we increase the stringency of the panel design, the off-target effects, off-target rates decrease significantly, right? You can see up to a 10 to 12% decrease in the off-target effects when we move the stringency from low to high. So depending upon, and it doesn't really affect the other metrics where the duplicate rates or the in 30x coverage or even the uniformity is not affected, right? So this shows that our panel designs are very efficient, and then we can actually play with the conditions right from the panel design stage to optimize so that you can get the best results that you're looking for. And the second aspect is the hybridization workflow, right? So the Twist fast hybridization workflow. First thing, it speeds up your entire workflow. You can finish the hybridization capture. The actual aspect of hybridization can range anywhere from 2 to 4 hours, and it's very efficient. And then it is actually customized you can customize it according to the capture panel that you're using. And in this hybridization workflow, the fast buffer that we use is more dynamic. So to say that, so if you look at the graph up here, which is with the standard wash buffers, you can see that even if you change the temperature from room temperature to around 70 degrees, after a particular point, there are no changes in the metrics, qualitative values that you get for the metrics like 30x coverage, which is shown up in green up here, or even the off-target effects that are shown up blue. And it doesn't really change the zero target coverage as well, which is the probes that are lost due to washing, essentially. But when you use fast buffer system, you can see that the wash temperature ranges from, you can actually vary the wash temperature from 60 degrees to 70 degrees. And you see, as you go up the temperature up here, you get a really good changes in the metrics. So the off-target effects reduce from at least 10 to 15% reduction in the off-target effects up here. And then the uniformity, irrespective of the change in temperature, the uniformity stays pretty even, almost to a very high temperatures up here. And after that, it suffers a little bit because we are washing away some of the probes that will cause a dip in the uniformity. But at the same time, the on-target rate does fluctuate. So depending upon what exact metrics we want to achieve, we can fine-tune the wash buffer system so that the results are what we are looking for. Whether if you want a higher sensitivity or low off-target effects. If you don't mind sequencing a little bit more and get higher sensitivity, you can actually do a little bit less temperature washing. And coming to the hybridization time, we saw how it affects the panel size. Actually affects how different panels get affected by different hybridization times. And here you can see that there is not much correlation between different panels and the hybridization time. By 2 hours, you almost get same kind of uniformity. Same kind of duplication rate. So that means the system is getting saturated right after 2 hours of hybridization time. So we suggest starting with 2 hours for any kind of panel that you're working with. And then depending upon the metrics that you get, you can actually adjust the hybridization time anywhere from 2 to 4 hours for improving upon the metrics that you get at the 2 hour time point. So the third addition, or the third addition in the hybridization workflow is addition of the Twist Methylation Enhancer. Right, this is a very unique and innovative product again from Twist, and these are actually special blockers that we add during the methylation capture. So here you can see when we add, depending upon the panel size, when we add methylation enhancer, it reduces the off-target effects pretty significantly. You can say at least 15% off-target reduction depending upon the panel size. But this methylation enhancer doesn't really affect the other metrics like uniformity. You can see it's a very tight distribution up here. And then the coverage at 30x or even the duplication rates. So this doesn't really detrimentally affect any other metrics, but it does significantly reduce the off-target effects that you get. And again, lower the off-target effects, better the sequencing data, and also significant cost savings. So now combining, the library prep and the Twist targeted enrichment, let's see how well the panel performs when we take it from end to end with the EM-Seq workflow, library preparation, and then the target enrichment. The first thing is, compared to the bisulfite conversion, the EM-Seq enzymatic conversion produces better metrics in almost in all segments, right? You can see the first important thing is reduction in these off-target effects, 15 to 20% reduction in the off-target effects. And the uniformity is increased. You can see up here, the uniformity goes up. The bisulfate sequencing produces less uniform target enrichment compared to the enzymatic conversion. And the coverage itself is significantly better with the enzymatic conversion. One thing that doesn't change much is the duplication rate. So this indicates that the enzymatic conversion is actually able to capture all the molecules, right? When so it is capturing all the molecules. It is converting, the library conversion converts all the molecules essential, and then we are using those captured molecules to actually get more uniformity compared to the enzymatic prep out there. And again, if we just look at the global effects on the GC, global distribution of the GC after the capture, compared to the bisulfite conversion up here, you can see that the enzymatic conversion has less bias. Over here, the x-axis shows the GC content, and y-axis shows the target read counts. You can see that the global distribution is pretty uniform when it compared to the bisulfite conversion, which shows a very bias towards the high GC regions. And to show this in perspective of the target capture, we used a 1.5 MB capture panel, which had both the hypomethylated regions and the hypermethylated regions. And here you can see on the top, the enzymatic conversion, which is green up here. The enzymatic conversion is more uniform all across the GC content, right? Irrespective of the GC content, you get a very uniform capture for the hypermethylated regions. And this is actually replicated when you're using the hypermethylated regions as well. The capture is pretty uniform. But when you look at the the bisulfite conversion method, it is significantly biased when it is capturing the high GC rich regions. There is poor, the efficiency is very bad compared to the enzymatic conversion. And this actually plays an important role because most of the regulatory regions, the CPG islands are part of this hypermethylated regions. And if you're not able to capture them properly, that will, you're essentially losing your data up there with this bisulfite conversion method compared to the enzymatic method. So we have seen that the enzymatic conversion, when coupled with the enrichment, it captures all the regions really well, and it is sensitive. But how do we define the sensitivity when it comes to methylation panels, right? In case of regular somatic mutations, we know that what depends on the sensitivity is when we are able to capture that 1% mutated region in compared to the high number of wild type molecules out there. But in case of methylation, the sensitivity is how well we are able to differentiate between low methylated regions and highly methylated regions, right? How well we are able to look at the differentially methylated regions. So to demonstrate this, what the experiment we did was we actually mixed various cell lines. We took DNA from various cell lines that has different methylation status, ranging from 5% to 100%, actually. And when we subjected it to enzymatic library preparation and target enrichment, you can see that irrespective of the methylation status, you get the off-target effects are not that significantly different. And you get very tight distribution of the duplicate reads and also very tight distribution of the uniformity. And the coverage is also very good, irrespective of the methylation state. And this is expected, right? And, it should not change depending upon the methylation state of the target. But the real data is how sensitive it is in capturing these differentially regulated states. So here in this graph, you can see that when we looked at cancer-specific biomarkers that show differential methylation states, you can see that we are able to capture all the molecules, and it exactly reflects the status of the methylation of these cancer biomarkers. You get very minimal methylation reads in where there is no methylation at all, and when it is 100% methylated, you get those reads. So this shows that the system is extremely sensitive, right? So this shows that we are able to capture all the molecules and then capture and at the same time saturate all the molecules during this conversion process. So which is, I think this is one of the most important things that you want when you're actually studying the differentially methylated regions. And just to put it in perspective, when you compare the same, efficiency with, compare the same thing with bisulfite treatment and the enzymatic treatment. So the graphs up here show that the percent offbeat is on the y-axis, and the different states of methylation are on the x-axis up here. You can see that enzymatic conversion, the off-target effects are far minimal compared to the bisulfite methylation, bisulfite conversion process up here, right? Irrespective of the methylation state that the target is. But the significant difference is at the uniformity of the capture. So if you see the fold 80 base penalty, which is on the y-axis up here, you can see that the distribution, it has a very tight distribution, the uniformity, irrespective of the methylation state of the sample, it has a very good uniformity. But in case of the bisulfite treatment, as the methylation state increases, there is significant bias for the regions that are hypermethylated. So this, along with the previous data, it clearly shows that the system is extremely sensitive. It can capture all the molecules, and then it can saturate all the molecules, giving you the sensitivity that is required for looking at differentially methylated regions, which is one of the key results that we aim for when we are doing methylation capture. And again, because you're using less amount, you're getting you're getting good library complexity. You can actually save, load more samples onto the sequencer, and you can save more per sample cost. And this is not, the question comes where, whether these metrics will trickle down to every panel size, right? So to address that question, we used 3 different panel sizes, ranging from 0.5 MB to 50 MB. And the system performs exceptionally well, irrespective of the target size that we have used. Here you can see that the off-target effects for all the 3 panels are less than 10%, with for the bigger panel, the off-target effects being around 7%, and for the other 2 panels with around less than 10%. And the uniformity is also really nice. It ranges from 1.6 to 1.5, showing that irrespective of the panel size, you can get really good uniform capture. The coverage is really good. It's more than 90% for all the 3 panels up there. And depending upon the panel, as the complexity of the panel increases, you can see a difference in the duplication rates up here. Seeing this, every custom panel is different, and then it does require optimization. And with the hybridization workflow that I talked before, you can actually optimize it in such a way that you can concentrate on the metrics that you are interested in, and then move forward with the conditions that will give you the best performance for your panel. And finally, looking at the competition up there, right? How well we perform compared to the competition. So for this workflow, we don't really have much competition up there. But so in this slide, I want to show you, how well Twist performs right off of the box compared to the competitor, where the data has been optimized. I mean, they optimize these panels for quite a while for achieving this kind of metrics. So you can see that there is 3 times better performance when it comes to fold enrichment. There is no comparison for the uniformity, right? We are miles ahead when it comes to the uniformity of the capture. And there is at least 20% reduction in the off-target effects, 15 to 20% reduction in the off-target effects. And the on-target rate is also very high, right? So all these things equate to one thing: you're recovering more amount of target regions that you're interested in. So you can put more number of samples onto the sequencer and get really good results, and with minimal sequencing costs. And looking at a little bit more about how well our system can get the DNA patterns. So these methylation patterns, in the human genome are emerging as key biomarker, right? Especially for when we are using circulatory tumor DNA from liquid biopsy. These methylation patterns can be used for monitoring the progression of the disease from early detection to late stages. So here you can see that the red lines are the normal DNA that we get from the patients, and not patients, normal DNA, and the blue line pertains to the circulatory tumor DNA. And when we use Twist panels, you can see how well we are able to demarcate between the methylation patterns, between the normal control samples and the cfDNA control samples. And these patterns are exceptionally, very important when you're looking at differentially methylated regions, because these using AI systems, you can actually clearly differentiate between early cancer states and the metastasized cancer states using these methylation patterns. And instead of looking at single methylated region, which most of the other assays out there look for, when you're looking these patterns on a holistic basis, this gives you better information and better confidence in actually classifying the stage of the disease. So in summary, so I have shown you the results that demonstrate the unique capability of the Twist technology and the Twist target enrichment panels for the methylation sequencing. And we show how well they perform across wide range of methylation targets for both hypermethylated regions and then methylated regions, and then how well we can optimize the workflow so that straight off of the box you can get the results that you're looking for. And when this combined with the efficient enzymatic preparation from NEBNext with the NEBNext module, you get really uniform libraries, and then you get more sensitive data, and then ultimately you can actually put more number of samples on a sequencer, and then you can actually save a lot of sequencing costs when you're using this system. And you can actually develop very high sensitive assays for the methylation needs that you are working with. So with this, I'll thank you guys for the attending this seminar, and I'm ready to take some questions. Thank you.
Thank you very much, Dr. Bommakanti, for the exciting presentation. So I see many questions coming in through the Q&A box, but due to time limitation, we'll quickly answer a few of them. So the first question, can we multiplex samples using this methylation kit? And if yes, how many samples do you recommend?
Yes, so most of the data that I've shown here is using single plex, but we recommend an 8-plex, and you should be able to 8-plex, multiplex 8 samples without any issue or without any effect on the metrics that you achieve per sample.
Okay, great. So the next question is that the Twist methylation detection system has the conversion step before the index PCR step. So this is different from the traditional methylation detection process, whereby the conversion step is after the index PCR. Can you explain the benefits of this new process?
Essentially, okay, can you repeat the question, Eli?
Okay, sure. So our Twist methylation detection system, in the protocol, the conversion step is before the index PCR, but the traditional methylation process is conversion steps happens after the index PCR. So what do you think would be the benefit of having this conversion step prior to the index PCR step?
So essentially, the index PCR is just for ensuring that you have barcoded individual samples, but the main effect comes from the actual conversion process. Right? When you're using enzymatic method, you are ensuring that the DNA is not damaged, and then you're converting more number of molecules. But additionally, it doesn't confer any real advantage just because you are adding indices to the modified sample.
Thank you. So maybe one last question. How does stringency affect the capture of partially methylated DNA molecules?
Oh, so again, it depends on how complex the molecules are, right? So as I showed you in the slide up here, let me quickly go back to that slide. So if you can see the slide up here, just because we are modifying the target, that changes the GC composition of the reference target. And once the GC composition is changed, the inherent structure or the repetitiveness of your DNA will change, right? So now we can, when we adjust the stringency, you are actually ensuring that so for example, if it is a very if it becomes a very low GC rich region, and if you wash at a very high stringency, there is always a chance that you're losing that probe. So depending on when you adjust the stringency, so for example, wash it at a bit lower temperature, you ensure that this low GC rich region or the high GC, AT rich region still is present in the capture, and you can look at that region. Right? So this mainly helps in ensuring that you're retaining all the targets because of this change in the GC composition compared to your reference target. That's the reason why stringency plays a very important role in how you analyze or achieve at the data that you're looking for.
Okay, great. There are many questions coming in, but due to time limitation, we will reach out to you offline to answer your question. So there's one last question that Ananth we would like you to answer here is why the enzymatic conversion method results in longer DNA fermentation patterns?
So again, as I showed you in previous slides up here, the bisulfite treatment is actually harsher. It does result in DNA damage, which chops up the DNA, right? But especially if there is nick on one strand, and when you're PCR amplifying, eventually ending up with a smaller fragment. But with enzymatic fragmentation, you're not doing any DNA damage. It's just modifying the DNA that you're looking at. And the DNA fragment length will essentially be the same that you determine using the mechanical fragmentation step. So it's essentially because there is no DNA damage, your DNA is intact, and you get better fragment size with the enzymatic conversion.
Okay, great. Thank you so much. So once again, thanks Dr. Bommakanti for the exciting presentation. And one last quick reminder to all of you. So following this webinar, you will be redirected to a brief survey. So if you have 1 minute to spare, we would greatly appreciate your feedback. And thank you so much for attending today's webinar. I hope that you find it insightful and informative. So have a great day ahead, and thank you so much.
Thank you.